Hi everyone, I'm Nilla, and me and my partner Jessica are going to be presenting on the phylum Rotifera. So first for our outline, we're going to be going over the body plan. Some of the specific morphologies for the anatomical features might vary from species to species, but they're going to follow the same general body plan. Then we're going to be going over the classes Eurotitoria and Hemirotitoria, and within those classes we have subclasses Monogenonta, Sisonidia, and Acanthocephala, and Doloidea from the root words rota for wheel and fura or to bear. So it's a wheel bearing organism. That word wheel comes from the ciliated corona at the head of the organism. You can see in this picture over here. And when those cilia beat, it resembles kind of like a rotating wheel. So that's where that name comes from. In these organisms, we have bilateral symmetry, and with that, we have cephalization. So it does have a head area and a tail area. It's triploblastic, it is a protostome, and it is pseudocoelomate, so it has no mesoderm and its organs are kind of suspended in fluid. They do have a kind of soft gelatinous body, and they don't have an external cuticle to really provide it structure and protection. So in order to get that structure, they have an intracellular protein lamina, and the thickness and flexibility of that kind of varies from species to species, but that's the general thing that you'll see in Rotifera. And another interesting thing is that their epidermis is there aren't cell walls in between, or there aren't walls in between those cells. So it's kind of just like one long fused cell with many nuclei. And so with this, we can guess that gas exchange probably occurs through diffusion because we don't have any organs that are specifically for gas exchange in these organisms. And they are pretty small, and seeing as they have no cuticle or any other layers of tissue, they can probably do gas exchange just through diffusion through the integument. Follow this body plan with three main regions: the head, the trunk, and the foot. Like I mentioned before, because it has bilateral symmetry, it does have that head region and a tail region. So in the head, it has a ciliated corona, so that's this part up here. The corona is mainly going to function for feeding and locomotion. So when the organism beats its cilia, it can draw water and food particles in, or it can push the water around it so that it can move through the water. Past that corona is the mouth and the mastax and the trophy. The mastax is basically the throat pharynx structure, and the trophy is going to be the jaws. And these are going to vary a lot between different species in terms of their morphology and the way that they will allow the organism to feed. And because of that variation, observing the trophy will help you in identifying what species of rotifer that you're looking at. We also have a simple brain, a simple bilobed brain behind that mastax, and it's going to bring in information from the eye spots, chemoreceptors, and tactile receptors. And most of these guys will have a complete gut, although some species have secondarily evolved an incomplete gut, so they've lost their anus. And they are also pseudocoelomate, so they do not have a mesoderm lining. And then we have the foot. The foot isn't always present. It's not present in the organisms that are mostly motile, that'll mostly swim or crawl. But for the organisms that are sessile or that will stay in one spot from time to time, it will have a foot and a toe or multiple toes that will have adhesive glands on them that'll allow them to stick onto whatever substrate it is that they'll stick onto. The class that we're going to be going over is Eurotitoria and the subclass Monogenonta. Between the species found in the subclass, there is a bit of variation in the environment that they're found in, the locomotion that they exhibit, and the morphology of their mastax and trophi. They are mostly freshwater, although some are marine, and they might be swimmers, creepers, or completely sessile. One method of feeding that is seen in a lot of Monogenonta is raptorial feeding. So while a lot of rotifers use their cilia to bring in food particles just by beating the cilia, some monogenonta or many monogenonta have greatly reduced cilia that aren't really going to be used for that purpose and are really more used for swimming. But instead, they have these trophi that have these long pincers, this picture down at the bottom, and they're going to be able to reach out and grab food particles. A somewhat unique feature of the monogenonta is that they have mictic slash amictic reproductive cycles. So they will undergo sexual or asexual reproduction depending on the environmental conditions at the time. So that's what this diagram on the right shows. What this is saying is basically that you will have amictic females, which are diploid females that can produce amictic eggs mitotically that don't need to be fertilized because they're also diploid, and they'll be able to divide mitotically further and create a fully formed amictic female. That process is called parthenogenesis, and that can keep continuing as long as the environmental conditions are ideal. As soon as the environmental conditions become inideal, the organism will detect that change and it will develop into a mictic female, which will undergo meiosis to create a mictic egg. And those eggs are going to be haploid. Those eggs will need to be fertilized by a male, and if the eggs remain unfertilized, then they will develop into males that will produce sperm by mitosis. Once the egg is fertilized, it will form into a diploid female, and that zygote will remain dormant until the conditions become ideal again, and it'll go back into that amictic cycle. The next class is Hemirotatoria, and the first subclass is Sisonidia. Sisonidia are epizoic on the marine crustacean Nibalia, so they live on its external surface and feed on its waste and eggs. Researchers also think that it can feed on the organism's hemolymph by piercing using its fulcrate trophi. So that's this picture down here. It'll pierce through its gills. And something unique about this subclass is that it's the only rotifer subclass that can only reproduce sexually, as far as we know. And the males are actually fully developed and diploid, and the females will only produce mictic or haploid eggs.
Okay, so this is the subclass Acanthocephala. They are nicknamed as spiny-headed worms. They're endoparasites, so they live inside the intestines of birds and fishes, and they aren't really known to infect humans. They're unsegmented worms that have lost their digestive tract completely, but they do still possess their reproductive organs. So this is how they're able to attach to their host. So they have a retractable hook, which allows them to have their body close to the intestines of their host. This is the reproduction and development. So males have genital ganglia that have sensory pores at the tips. Females have gonopore, vagina, and a uterus, and both male and female reproductive systems are in ligament sacs. Once a sperm fertilizes an egg, acanthor larva is released. Embryos that are still in early stages are pushed back into body cavity and continue to develop. The larva that was released have to look for intermediate hosts to continue their life cycle. This is the subclass Bideota. They live in dry environments, freshwater, moist soils, and they're either marine or terrestrial. They are entirely females, and they do have a developed corona on top of their retractable heads. So they go through asexual reproduction. So males are not needed and they are unknown. And then once they do reproduce, they produce only females. This is their behavior. They're either sessile, they swim, or they crawl. And then they're able to feed by using their cilia that is on their corona to make water flow into the grinding mastax. These are the questions for our phylum. Phylum Rotifera, body plan consists of blank, blank, and blank. Which structure can be most helpful in telling rotifer species apart? These are the answers. Thank you.